So the leukocytes, basically the innate immunity is mediated largely by granulocytes. Adaptive immunity, which is mediated by lymphocytes, and there is a growth and the development and the activities of it, because the mature neutrophil, eosinophil, basophil, with the different types of staining properties, you can observe these cells under the microscope, and the development, activities, secretion of these granulocytes and lymphocytes, they are interconnected and very often cooperative. As one is not working properly, the other has to come up and do the activity of that. So neutrophils, which have a polynuclear system, and it has a amoeboid type of system which engulfs the cells. There are more amount of neutrophils in the body. As we can have a WBC enumeration, we can maintain or we can enumerate the WBC, because when we see any of the pathology report, when we go for this CBS type of testing of blood, we know that the doctor will tell you when any of the fever or any infection is there, then they ask you to do the CBC system or the CBS system. That is, you have to count the blood cell, and the normal blood count we have to give. And what is the normal blood count of WBC in the human? Normal. What is the blood count of the normal human WBC count? Cubic millimeter. It is 11,000. From 4,000 to 11,000 per cubic mm is a percentage of the WBC in the blood, and the RBC is 4.5 to 5.5 million in the normal. What is the percentage of hemoglobin in a normal blood? Hemoglobin percentage. Why we have to check the hemoglobin percentage of the blood? Anyone know what is the hemoglobin percentage? कितना रहता है? For the male it is different and for female it is different. Yes. So what is the percentage of that we calculate? Basically the hemoglobin percent in the gram percent. So for the male it is from 11 to 16 basically. For the, it's different for plus minus 1 range, and it is from 11 to 16 is for the normal, is for male, and for women it is from 10 to 14 is for the female. And if it less, but most of the Indian women, they are lack of or less hemoglobin percentage, and therefore most of the women of India are anemic. Anemia is there, a disease where the hemoglobin percentage is less. So these all blood cells which are there, the platelets, the WBC, RBC, has a specific amount because they are present in the blood, and blood has a function to carry the oxygen as well as the food material to various parts or various organs of the body. For the vital functioning, blood is very important, and it should maintain the pH, it should maintain the various type of cells at a specific level. So these lymphocytes, they have a certain type of system or basically a structure, activity and the function which is responsible for generating the immune response. So neutrophils, which are 60% of the WBCs, and they patrol the tissues, because patrolling the tissues means they are acting as a soldiers. They are moving all over, seeing whether the infection is there, whether anyone is coming. So they are patrolling continuously the tissues, and they squeeze out of the capillaries. They come out of the capillaries, they see whether the infection is there. They are at a very, so like the soldiers which are there at the line of controls. Like defense system, a military system is like the soldiers are there continuously, they are at the border, and therefore we are sleeping properly in night, we are following our dreams, and these soldiers are playing a very important role. They are not made difficulty in any of the conditions, like the weather condition or any calamity is there. They are continuously doing their duties likewise, these WBCs also have the property because they are also the soldiers of our system, of our body, and continuously they are patrolling to the tissues. The large number of cells, they come and they inflammate at a infection site and release a certain amount of chemicals, and these chemicals are responsible for taking this material towards it and basically digest that micro any of the infectious issues. So dead neutrophils also, which make up the large proportion of the pus, is also to be phagocytosed, because if the neutrophils are working and then they get killed or die there, because it has a fate, every cell has a specific amount of fate and there has a life cycle, and when it gets die off or gets killed or die, then there has to be clean up there. So the pus cell, if the pus formation or the pus cell formation is more there, then the infection is causing that. That means it is not killing. So dead neutrophils, they make up a large amount of proportion of the pus, and therefore they has to be removed by different types of cell. Then macrophages, they are larger than the neutrophils or neutrophils, and they are present in the organs, not the blood. These macrophages, they are basically in the bone marrow as monocytes, and once they reach the organs, they have a specific type of phagocytosis system, and they initiate the immune response. They display the antigens from the pathogens to the lymphocytes. So these macrophages or microphages, they have a property which once they reach the organ, they have a property to initiate the immune response. The first cells which are basically generating the immune response, because we are now seeing the primary line of infection, the skin, the mucus is the first line of defense, but when it surpasses from that and when it comes under the cells, then the macrophages are very important, and these macrophages, they are basically larger than the neutrophils and very active, and they are near the organs. They are not present in the blood. They are there in the organs, they are in the bone marrow, they are acting as the monocytes, and they basically initiate the immune response as they display the antigens from the pathogens to the lymphocytes. Then bacteria, they are phagocytosized into vacuole, which we call as a phagosome, then phagolysosome, which was of the balytic enzyme. This is a mechanism of the phagocytosis. So process of phagocytosis, that is phagosome is forming, lysosome is there, and the damage and the digestion of that, because release of these microbial products or basically the lytic enzyme destroy the bacteria. Brucella, leprosy, salmonella, cholera, many of these bacterial infections are basically seen by these type of phagocytosed cells, or we call them as a macrophages or monocytes. Then another types of subsets, as the macrophages are there or the monocytes are there, then we have a lymphocytes. We call them as a lymphocyte sets or lymphocyte subsets, and the two types of lymphocytes, we call them as a common lymphoid precursors, because these are from the lymphoid system. Lymphoid precursors and lymphoid system is there. So this lymphoid system is basically a dendritic cell and natural killer cells. So we have two types of lymphocyte subsets, and these two types of lymphoid subsets are T cells and B cells. Now T cells, which are from thymus T, they are from the thymus, and they are activate B cells and macrophages, and they are T helper cells. So we have T cells of two types. One activate the B cells, and they are known as T helper cells. So I want the classification from you people. You have to give an assignment of what are the different types of lymphocyte subsets of T cells and B cells. So what are dendritic cell, what are natural killer cells, what are T cells, what are B cells, T helper cytotoxic T cells and plasma cells which are producing the antibodies. So we have T cells and B cells. T cells are again classified into two, that is T helper cells and cytotoxic T lymphocytes that kill viruses and infected cells, because again there is a classification, because these T cells, they do not have the property because the surface antigen or surface material is there which have a property to kill specific type of cells only. So activate P cells and macrophages which are T helper cells, and CTL, that cytotoxic T lymphocytes which have the property to kill the viruses. So T cells of TH we call them as the T helper, and CTL we call them as the cytotoxic T lymphocyte. We have a dendritic cell, we have the natural killer cell, and another is the B cells which we call as the antigen presenting cells because they produce the antibodies. Once the antigens are presented over that, then they form a specific antibodies for that antigens, that is the complement fixation, and we have that MHC1 and MHC2. So natural killer cells or NK cells, they are leukocytes and they are white blood cells. We have two types again, of granular and granular. Lymphocytes, monocytes are granular, whereas granular are basophils, neutrophils and eosinophils. Agranular are T cell, B cell and NK cell. So natural killer cell, B cell and T cells are agranular one, whereas granular are basophil, neutrophils and eosinophils. Then we have a specific type of cells like the immunity and the cancer. So I want again a note from you. How can you relate immunity and cancer? So because cancer cells, how they have, because they are again no discrimination of cells. So how the cells form, how the cancerous cell formation takes place, and what immunity plays a role for formation of this cancer cell. What happens to immune cells and the cancer cells? So what is the fate of immunity and cancer cells? What is the role of these natural killer cells? Natural killer cells are a type of cytotoxic lymphocytes which constitute a major component of the innate immune system. So they are NK cells, we call them as the natural killer cells. They are from the monocytes, they are agranular, and these NK cells, they have a major role in the rejection of tumors and cells which are infected by viruses. The cells get killed by releasing small cytoplasmic granules of protein called perforin and granzyme that cause the target cell to die by apoptosis. So these natural killer cells have a property to basically kill the cancer cells which are either forming by the bacteria or the viruses or the tumors which are forming, but these are the natural killer cells and they kill these cancerous or tumor cells which are forming by these performins and granzyme, and they cause a target cell to die by suicide, or they apoptize that the cell directly have a property to get killed by itself. Now the role of inflammation, because it's very important in the immunity. If any of the infection occurs, then the inflammation is there. So inflammation is signaled by mast cells which releases histamine. Now histamine is a amino acid like or histamine, which is a product or carboxylation product of the histidine, and histamine released from the mast cell has a property to inflate that. So histamine causes fluids to collect around an injury to dilute the toxins, and this causes the swelling. So how the inflammation occur? This is a process of, because histamine is very important, which causes a fluid to collect around an injury to dilute the toxins, and this causes the swelling. And the temperature of the tissue may rise, which can kill the temperature sensitive microorganisms. So it is a process, or basically the inflammation property, how the inflammation occurs. So inflammation and immunity plays a very important role, because what it's a process when immune response is generating, it has to inflate or inflammate. So there is an inflammatory response which in the body, which is the second line of defense basically. The first line of defense is, I told you about the microbial flora. The inflammation is the secondary or the second line of defense against the invasion. Once the infection is there, then it swells. So the inflammation is there, and the inflammation is due to certain type of chemicals that is secreted, like the histamine and certain type of enzymes it release, and it swells at the type of infection. So there is tissue injury, there is irritation, there are arterioles constrict initially and then dilate. It slow the blood flow and it slow the margination of the leukocytes. That is, the margination of the leukocytes means it gets marginalized. It goes around a specific type of system or epithelial layers, and leukocytes or the blood flow gets slowed down because of the margination of the leukocyte. It escapes into the tissues by diapeptides and accumulate in large numbers. So there is inflammation means there is a tissue injury, there is irritation, there is certain chemicals are forming inside, and the arterioles in the lungs constrict initially and then they dilate, and it slows the blood flow, and leukocytes marginates and escape into the tissues by diapeptase process, and it accumulates in large number causing the inflammation. So inflammation is basically an injury. Sometimes the inflammation is due to heat, the redness is there, the swelling is there, the pain is there, and the loss of function. So the inflammation has a progressive effect. If the inflammation is there, then the type of burning sensation is there, that is, heat is there, burning sensation. Then the redness occurs if there is the infection. Then certain skin shows, it's a defense line, first defense line, it shows a redness over the skin or wherever the infection is there, and then it gets bulged because it dilated, so the inflate or the swell, and after that it pains. So and finally it lost the function of any of the organ. So that is a process of inflammation, which is a secondary line of the infection. So it output the plasma and dilute the toxic material. It produces the fibrin barrier and localize the infection. So there is a process of inflammation. Fever is very important because natural defense mechanism destroy infectious agents, then leaves fever, or we have a hypothermic type of process, and that produce or secrete certain type of interferon, certain type of infectious agents. This is again a natural phenomena, which if any of the fever or any infection or inflammation is there, then fever comes. So there are certain acute phase proteins where the infection and injury produce, and these are the CRP. Nowadays also we are seeing that CRP proteins, that is C-reactive proteins, are very important, and they also show that if these proteins activates, then through this the host defense system, it has a preventing the tissue injury and repairing the inflamed lesion. So CRP, that is a C-reactive protein, is basically a biomarker of finding the inflammation or the infection rate. One minute. My internet connection slowing down. So thus the cells have a property to get into inflammation, and that basically a second line of infection or the immunity is important when we see inflammation. And then different types of cells, different types of organs, and all those constitute to generate the immune response, the first line of defense and the second line of defense. So cells are for the second line, that we call as a cellular immunity, humoral immunity and the first line of defense, where the organs are responsible for generating the immune response. So we have to classify, basically this is an introductory class of immunology, where how the organs and the cells have the property to generate the immune system or the immune response. So this is basically a introductory part. Then you have different types of cells, different type of neutrophils, phagocytes, cells like monocytes, macrophages. They have the property to generate the or develop the immune response. So for immunology, you have a first line of defense and you have a second line of defense. So second line of defense, the cells are constituting the second line of defense, whereas in the first line of defense, the organs which are responsible and the natural phenomena like the microbial flora and the process of urination, respiratory tract, there are certain organisms, there are certain chemical secretion, and they engulf them and they kill the microorganism. So it's basically a introductory to the immunology. The detail that we have to see is various types of organs, because specific organs that we have to describe how these organs are working and what are different types of cells that are working to have a immune response generation. So for today's, this is second line of defense and first line of defense we have completed, and yesterday I told you about what are different types of system, or what is innate immunity, what is acquired immunity, adaptive immunity, and how it is forming because of the genetic constitution. So this comprises basically the introduction of the immunology. So you are getting me all, students? You have an idea, basic idea of immunology? Okay, so I will give you all the notes, and on Monday at 4 or 3 again I will be giving you the link, and we are taking a detail of these antibodies and how these various types of antibodies are working for the B cells. So how the secondary response of the B cells takes place. So basically the T cells and the B cells. So cells in detail we will be dealing with in next class. So I will again informing you about this. Akshat, you take the attendance in the class. Okay. So any difficulty? Yes. You can ask the questions. Next week I will tell you, I will give on the WhatsApp when you have to submit, because you have to give again online on the Google Classroom only. So I will tell you, but 1 week time is there. So in next week, I will give you, in next week I will give you date when to give. And after this completion of this immunology, both the units, I will be taking your MCQs on all the 4 units. So 2 units that I have completed, that also you have to prepare the for tests, or I'll taking your, yes, I'll be sending you today itself or tomorrow. Okay, thank you all. Thank you, take care. Thank you. Yes, thank you. Yes, thank you all. Thank you.